Okay, so what I would like to discuss today about is two aspects of tissue morphogenesis. The first one is how forces are produced at the subcellular level to drive cell shape changes and yield microscopic tissue changes during morphogenesis. And second, how the adherent junctions are organized. And these two aspects, I think, are important to understand cell behaviors during tissue morphogenesis. And this morning we've heard that indeed it's important to look at this intermediate scale between genes and macroscopic movement. So what I would like to put emphasis on are quantitative measurements, how we can measure properties such as tension in tissues, measure the organization of adhesion components in cells, in vivo, and in the context of this course on quantitative approach to tissue morphogenesis, I think it's very important to be able to connect key parameters, so basic mechanistic parameters to predict quantitatively cell behaviors. And I will try to show you with different examples how we can measure properties, mechanical properties, and as well organization of structures during tissue morphogenesis. Since I know that I might not go to the end of my talk, I would like first to thank people who have contributed to the work. So I will talk about Matteo's work when he was in the lab, in my group, and as well in the group of Thomas. I would like to thank Madhav Mani, who's a postdoc here at the KITP, who contributed to the second part of my talk. And I would like to thank as well Thomas, who I've been collaborated with during the past 7 years. So in case I don't reach the end, it's done. So, and I will as well mention people who are not here in the audience and who have been doing the work. So to start with and talking about cell shapes and cell shape changes, I would like to show you this very nice and striking example of organization of cells in a tissue in the retina of the Drosophila, where you have striking patterns which are very regular with this unit, this I unit, which is repeated.
Very pretty. I was saying they're pretty. It reminds me of tiles.
Yes, this is very. And these units are repeated about 900 times in the eye. And the organization of these cells is remarkable. And what is striking is if you compare the organization of these 4 cells, which are called cone cells and which are surrounded by primary pigment cells and secondary pigment cells, the organization, the cell shapes resemble the organization of soap bubbles. So if you take 4 soap bubbles of the same size, I think this experiment is quite hard to do. I haven't done it myself, but at least if you prepare correctly 4 bubbles of the same size and put this on the substrate, you get this shape with angles very well defined, and they are very similar in terms of angle, organization of shape, as well curvature, to these 4 cone cells in the retina. So it's striking to see the resemblance. I would say that what we learn from this resemblance is that mechanics of surface is important. What we do not learn is what are the properties, at the molecular, supramolecular level, which yield to the property of cell surfaces and then which govern cell shapes. And in the case of these specific cells, cone cells in the retina, it has been shown by Carthew and Hayashi that the length of contact with these cells is determined by the relative ratio of N and E cadherin to some extent, and that N cadherin is present only between these 4 cone cells, but is absent of at the contact between cone cells and primary pigment cells. And if you remove N-cadherin between these 4 cells, you decrease the length of contact between these four cells, as it is shown here, and you change the geometry, especially you decrease the length of contact and change the angles between cells. So at least this suggests that to some extent the level of cadherins regulate cell shapes. But what is, in fact, the resemblance is striking, but if you look more carefully, already you see that here there is a bond. So there is a junction here which might play a role, and if you think that it's going to pull on this cell contact, you might expect a different organization. And indeed, if you would be able to make not only these 4 bubbles, but as well big bubbles around, you wouldn't get this organization. And if, yes?
Also, like the 3D shape of the cells, is it spherical or not really?
No, it's not at all spherical. I mean, if I would have shown you the organization of these omatidia, so you have below other cells, so it's very elongated structures. But still, the adherent junction is very limited. So you could say that the shape is determined in 2D. Okay, but what has been shown is that if you take into account, this boundary, which is presumably tense, even if it has not been measured, you would expect a different configuration with in fact, a junction in the other direction. So something is missing, and it has been recognized by different groups, our groups and others, that cortical tension is as well important to understand cell shape. So the extent of contact between cells is determined not only by adhesion, which tends to promote surface of contact between cells, so it promotes the extension of this contact, but the cortical tension as well is important and has an inverse effect. It tends to reduce the surface of contact between the cells. So now if you try to understand how cell acquire the shape, you have these 2 systems which are important, which at first approximation can be like split. You can think that arctomyosin network has a role on cortical tension, and adhesion cadherin molecules have a role in this, the extent of contact in adhesion. But what has been recognized during the past years, and I'm sure that Carl Philipp Heisenberg will as well talk about this, is that adhesion molecules, especially E-cadherin in epithelia, supports as well the coupling. So the coupling between the tension which is built in the cell to the neighboring cells. So the E-cadherin supports coupling, transmission, and as well resistance to forces produced in cells.
I just want to this picture that usually assumes that the tension is the same at the interface and in the rest of the cell, but it's not the case. I guess because it's been shown enough.
Sure.
Tension is not always strong enough to produce any force.
I agree. I mean, it's just as an introduction is to say that starting from simple rules of adhesion is not sufficient. Put tension in addition, but then how the extent of contact is determined in some system depends very little on adhesion molecules themselves in terms of quantity. But this is something which is not completely solved. But at first approximation, you could think these are 2 forces, like a weighting force, and here a force which resists the expansion of a contact. But what makes the system complex is that adhesion molecules themselves have a role in organizing actin networks, signaling actin networks, and as well supporting forces, transmission and resistance. But I'm sure there will be as well other talks talking on these specific aspects of cell contacts. But let's continue. So what I would like to, so the system I will discuss mostly during my talk is at least the system in which the experiments I will show have been done is the early Drosophila embryo. So Thomas has already shown this movie where you see the extension of the germ bond. So at the microscopic level, you have the elongation of the tissue along the antero-posterior axis. So this is the microscopic system we are looking at. At the intermediate scale, you have very complex dynamics that I'm sure Thomas will discuss, how actomyosin networks flow to our junction to drive cell intercalation. And what I would like to present today are our efforts to measure mechanics. So here I show an example of dissection. I will discuss more during the next slides. How we can measure mechanics, forces driving cell shape changes in this tissue, and as well how we can look at organization of cells contacts in this tissue and how, for example, adhesion changes over time and which key regulators change the organization of these structures. So first I will discuss in the first part how we can probe the mechanics of cell junctions, and second how E-cadherin organize at cell surfaces and how they are regulated. Okay, so this you've seen already, the early Drosophila embryo in which you have this microscopic flow of cells and elongation along the antero-posterior axis. And this is the time going on, typically 30-40 minutes to have this twofold elongation. This process is driven by cell neighbor exchanges, cell intercalation, and if you track cells during this process, you will see that cells which were in contact at the onset of elongation, here the green cells or the blue cells or the magenta cells, they lose contact, so the green cells are not in contact anymore after elongation, while magenta and blue cells gain contact during the process. And at the subcellular level, at least at the junction, at the level of junction, this process of cell neighbor exchanges is mediated by myosin II, which is anisotropically distributed in the plane of the tissue. You see that this vertical junction, so oriented along the dorsoventral axis, have more myosin, are more enriched, so there is more myosin along these contacts compared to the transverse contact. And this yield shrinkage of this junction, bringing these 4 cells into contact, forming a 4-way vertex, and then a new junction expands. And this is called a T1 transition, so exchanges of cell neighbors. And if you look at this distribution, you can ask the question whether myosin modulate contractility, modulate cortical tension, and then yield to drive cell intercalation per se. So are these motor act directly at cell surfaces to change the mechanical properties of the cells and drive elongation? So a few years ago, or about 5 years ago, we started with very simple model in order to understand morphogenetic movements. Other groups have used similar models which are based again on the physics of soap bubbles, or soap bubbles in which you would, in addition of the surface tension which determine the shape, you include elasticity. I will not go into the details, but here what is important is with this type of model where there is in fact no time, just mechanical model where you look at the balance of forces, if you assume that at some cell boundaries, so at this junction there is more tension than compared to the transverse junction. So in this case, there is 70% more tension along this contact compared to the transverse one. And if you ask whether this system will change in shape, you observe that by energy mobilizations, so by imposing mechanical equilibrium, it gives rise to an elongation of the tissue. So starting from a configuration as observed in vivo and imposing anisotropic tension, you observe shrinkage of junction, cell intercalation, and expansion of the tissue. So this suggests that anisotropy of tension, which that we think we can show that is determined by Moisin II distribution, drives cell intercalation and at the microscopic level tissue elongation.
So towards the end, I see very little vertical edges, and they are not red.
Yes. So in this simple simulation, we imposed only at the onset that vertical junctions are more tensed. If you would impose all the time that any vertical junction close to the vertical would be red, at the end you would end up with a single line of cell. Yeah. Does it answer you?
In reality, it is true that occasionally there's a fair number of junctions which were transverse at the time became vertical after some
Yes.
so.
Yes, and here we just imposed initially this condition of vertical junctions being more tensed.
And
If you
means when the edge undergoes the T1 transition,
then the new expanding junction here in this simulation have the same tension as the black ones. So we do not impose any constraint or any additional tension to drive elongation of a new junction. Yes.
Yeah, an edge effect on your model. So at the very end,
if you look around the perimeters, there's a lot of
residual red.
Here, you mean here?
No, the perimeter of the whole model.
Yes.
Red,
here,
red.
I agree. But here in fact, we didn't take attention very much. I could have shown you only this part. The tissue is the thing is that the tissue is large enough. You can impose. So to answer the question, if you impose these conditions only in the central part of the tissue, you have as well elongation, even if the other part will remain black because there is no constraint.
One of the things that just struck me was that a problem that we used to have in thinking about this was always to ask why germ band extension stops, why it undergoes a certain elongation, a doubling, and then stopping. And the way that we were thinking about it at the time would have just gone on forever.
Sure.
And yet, what you've done here by having the polarization event be a short little interval in time where you set it up and you say, okay, everybody check your neighbors and be unpolarized, and then let the system go, but don't allow any new cell communication. You are going to get a certain behavior that'll be a certain length, and then it'll stop.
I agree. I agree. It was okay. In these simulations, indeed, we imposed this very short time for polarization, but yes, and it works. Yes, and if you let the polarization over a longer time, then you can elongate more. If you let it forever, you might even which is okay.
And I think the interesting question is, so even which you talked about in Thomas and others, somehow control this polarity. Now, that pattern is very exquisitely controlled to start with, but will individually be somewhat modified. And the question is whether this collapse, the hypothetical collapse of polarity is a consequence of modification of the geometry.
To be really explicit, in the Irvine and Ken's paper, where Ken and I disagreed, was that Ken actually talked about a group of cells, the Eve-expressing cells, by differential adhesion, effectively rounding up And so the way he got the germ band to stop in his cartoon models was to have a big enough domain that would go from being a long rectangle into a square, and that was because he was treating only certain certain differences as being privileged. Whereas I kept wanting to have every interface be different from the other, but my problem was that germ band extension would go on forever. And so yours is a really interesting compromise resolution.
Yeah, this indicates that if you depending on the time over which you will set this condition of polarization, you can change the length at least.
When the previous kind of experiment where you showed the intercalation, where you marked the cells, and you probably know this, I'm sure that all these papers it's all described, but there's division going on during extension.
No, there is no. At least in most of. If I show you the movie again, you have. Okay, here you don't have division, so the number of cells is constant. Really, at the end of elongation, you do see division, and you will see here. If we go to the end of the movie, you will really see at the end in this region, you will see few cells dividing, but it's really at the end. Okay. Okay, so this is a model, but for any model, you need to. It's a model. You need to test it, and now I will discuss a bit the methods we developed to measure this tension, and which have been as well used by different groups with different systems. So here I would like to point out that we used near-infrared femtosecond pulses to break actin networks, and the rationale of this is to be able to really produce local plasma which breaks actin and which produce very little collateral damage, actin and structure which dense close to the membrane. And here is one movie where at the E-cadherin labeling, so you break the contact. In fact, you don't break the membrane, you break actin, as I will show you, cadherin which moves apart with actin, and then you can see the relaxation of cell contact after dissection. And I think you can do the same type of experiment during the next days with the 2-photon microscope, the 780. You just need, I suppose, to use the FRAP module, putting a bit more of laser that you would use for imaging, and you can try to do this type of experiment. From this, so you can see that actin redistribute after a dissection from the point of ablation. We control that the membrane was not broken. You can look at cadherin molecules, and again, so you see the breakage, redistribution of cadherin, and then relaxation. And what we learned from this type of experiment is the tension, at least the relative tension at cell boundaries during intercalation. So if you measure the speed of relaxation just after dissection, it's a proxy of tension at cell contacts. So the speed, the maximal speed of relaxation after ablation is given by the tension over the friction, and we don't know the friction in this case, but we can use the speed as a proxy for tension. And because we break this bond, we do see the movement of the vertex, which is pulled by these two remaining tension. So, yes? Sure? Yes?
Are we sure that there isn't sort of a shorter time scale? That's right. So presumably, at very, very early times are rather crucial, right? And the question is
how early it is. At least at least what we probe is like the elastic response of the system just after breakage. We haven't looked. Yes.
Okay, so here it looks like imaging at
Here or less than second. We can go below 1 second. Yes, in these specific experiments, at that time it was 2 images per second, and we can go to, let's say, 10 images per second.
Difference between 2 and 10.
No, I have to check. Okay.
I think there are two phenomena going on. One is the redistribution of actin myosin within an otherwise transiently, stable junction in terms of its length. And I think this phenomena we probably don't have the right resolution to resolve yet. But if you look at the kinetics of the junction relating to the whole material,
but still,
that is long time scale in this particular result.
Still, I do not fully agree. I mean, if you look even at these old images which were taken by Matteo a few years ago, we had you can see the redistribution of cadherin, which in fact doesn't go as really as fast as at the moving of yes.
At a naive level, both tension and friction depend on the thickness of the cortex.
Yes.
So if you have a change in tension,
I agree indeed. So that's why we yes, I agree. So that's why we try to do other types of experiment to distinguish the two contribution. At least this is the way we've done, and that many groups are doing still currently. And this hidden, this parameter might be very important. It can depend on the geometry of the junction, the thickness of the cortex. So I fully agree. At least this is how many groups, and still many groups, are doing. And hopefully we can do better by other ways. So when we've done these experiments at different time points during a junction shrinkage, so we took different junctions during cell intercalation. So before at the onset of elongation and during the shrinkage of junction. And what you can see when the junction shrinks, the tension, as revealed by the speed of relaxation, increases dramatically by 5-fold, typically. And the natural question, to test the model of anisotropy of tension driven by myosin 2, we did experiments where we inhibited myosin 2. And if you do this experiment, you observe that the relaxation of the contact after ablation is very, very slow compared to the wild type situation. So this indicates that indeed myosin 2 is responsible for the tension in this junction during tissue elongation. Now if you want to address the local balance, and I know now that there are methods, which are used to get from the geometry of junctions the tension in a given tissue. In fact, we made these experiments to check whether the balance was indeed true, and we made different cuts for different orientations. So during junction shrinkage, you have this junction which shrinks, and during shrinkage, this angle reduces. This goes along this direction. And if you compare the tension along vertical junction with respect to transverse junction, you do observe this local balance, mechanical equilibrium, and at most, when this angle is small, so when the junction has shrunk a lot, then you have about 2-fold differences, which says that these guys pulls twice more than these two guys when the angle is very small. So this indicates that anisotropy of tension of 2-fold is sufficient to drive cell intercalation, and from what we've learned from the model, the elongation of the tissue. Okay, so this was like a steady state view of cell intercalation and tissue elongation. And I would like to show you this movie, which to some extent not to some extent, which shows that the process of junction shrinkage cell intercation is very dynamic. And I'm sure that Thomas will talk about this later, presumably in 2 weeks, about the dynamics of actin-myosin network driving steps of shrinkage of this junction. Here you see myosin 2 in green, E-cadherin in red. So there are fluctuations both in myosin 2 channel and in cadherin. And so the process is much more dynamic than the process I've shown you as a constant shrinkage of junction. And if you see how a junction change in length, how the deformation is produced, you see very diverse modes of deformation. And we thought it is very important to rely on methods which really measure tension more directly to know what are the magnitude of forces which drive the bending of this junction, how much a change of forces produced in a cell can change the shape of the neighboring cells. So this is why we went for other types of mechanical measurements. And recently in the lab we started to do optical tweezers experiments with beads. So we can inject easily the beads in the Drosophila embryos in the CCSL stage, and eventually there are beads in the embryo, and then we can manipulate them and produce forces in vivo in the tissue. So here is an example of a bead in the cytosol that we can move. So we trap it. I will just briefly show you how we can do it. So the principles of optical trapping for those who don't know. Here we move the trap, so you can measure viscosity. You can do many things with these beads. And here we can start to do mechanics. So here you have a bead, 500 nanometer in diameter. You trap the bead, and you push the bead against the junction, and see how it deforms. And you can do it at different frequencies. Slow, it was fast frequency here, second time scale, slow frequency, and you can do radiology. At least you can think of doing radiology. We haven't done it completely, but we will presumably do it. So the time scale here, this was 2 seconds. This was 2 seconds, and these oscillations. And this is 10 seconds, this one. The thing is that it depends on the tissue you're looking at. So if you're looking at this germ bond or the mesoderm going so fast, it's true that if you want to do it very slowly, it's a problem, because then the cell will change shape during the process. But I would say you can do 1 micrometer of deformation over 10 seconds.
If you bring a bead next to the cortex, to the side of the cell, in principle, in the best possible imaginable world, how many seconds would you need to measure the tension?
So I will show you. I don't need to do this that way. We are doing the measurement of tension this way. I will tell you how we do it. Okay. The thing is that if you put your bead and wait too long, then the bead will escape the trap, because the forces of the tissue itself is very large compared to the forces that you produce with an optical tweezers. So the typical forces you produce with this type of tweezing is at most 100 piconewtons for 1 micrometer bead. So these are not huge forces, but still they are sufficient to produce. I can show you this movie again. They are sufficient to produce, to deform the cells. Yes. So these beads are really, we do not treat them. They are they are carboxylated beads, and yes, so we can move them freely. So sometimes they are stuck. This is true. They some are stuck. And
I think you told me that already, but there's a distance you cannot approach, right? So you can
this depends. So in this case, we do see that the beads even seem to enter the membrane in this case where the movement was, I mean, if you look back at this movie, it seems that there is maybe a region where the membrane is deformed before the bead enters it. Okay, so it might be a way to, measure cortex size as well, but. Wait.
The motion is complete control like a laser.
Yes. So
you get the time scale determined by
Sure. So
you manually.
Yeah. So maybe just for like since we are in a course, I think it's sorry, Boris. The bit. I mean, this is the sensitial. This is the. I mean, the beauty of Drosophila. You know, there are things which are not as nice in Drosophila, and. So you can inject in the syncytium where you have no cell. You inject early, and say by chance you have beads there. So you cannot inject all sizes. So that's. I mean, we would like to have larger beads, but then we get into problem. But it works. So, and it's not totally trivial. And to get the beads at a good plane, it's. At least it's feasible in the Drosophila embryo because of the syncytium stage. And so just for those who are not into optics, so you are doing microscopy during these days. So the optical tweezers use light to produce force. So with light you can push beads because photons are reflected, so there is a momentum which pulls the bead in this case because of the reaction, Newton principle, and you have as well refraction of the light, and this photon will be refracted this direction, so this will give rise to a transfer of momentum of the bead, and depending on the intensity of photons, if you have more photons here than there, you will have a net transfer of momentum which will bring the bead into the center, let's say the focus of the trap at the highest intensity point. So, and the position of the bead with respect back to the trap give you the force you apply. So it is really an easy way to measure force and to exert forces.
What is the set resolution?
You don't. I mean, usually this trap is like you have a given stiffness in the plane, but you have as well a stiffness along the optical axis, and this is very low along the optical axis. So you don't have much resolution. I mean, the bead is there, and it is when you move in the plane and you measure the distance between the trap position and the bead to get the forces. There is no real. I mean, the resolution is given. To answer the question of the resolution, is given by the focus of the laser beam itself. Yeah. But you cannot do force measurement along the optical axis. This is usually you can't.
Right. How much control do you have?
I mean, it's like a laser. You would change the focus. Yeah. Okay, so what we learn from this type of measurement, so this is the position of the bead with time when we do this this framing experiment. So this is the bead. It enters the membrane at this point. So the trap goes this way, but the bead doesn't follow. So the difference in position between the trap and the bead gives you the tension. So gives you the force exerted by the junction. So here, if you now, I mean, this is very simple model of the junction as a string that you would pull, and if you deform the junction by this force that you can measure, you can get the tension. And from the deformation, from the difference in position of the beam, you can estimate the tension at cell boundaries, and in this case it's typically 100 piconewton. So coming back to germ bond elongation, we've done some measurements, not many so far, but already we can see that tension at cell boundaries, depending on the orientation of the junction, is anisotropic. So this is the tension along vertical junction, and this is along horizontal junction. So it's a bit less than twofold difference, but there is a difference. And what is important in terms of numbers, so 100 piconewton, which drives this deformation of about 500 nanometers, not that much, but still already cell shape changes. 100 piconewton is, one motor, molecular motor, produces 5 piconewton force. So that this...
Let's speak force. Average force is much slower.
You mean average force? Yeah, sure. Yeah, sure. Installed force, okay. No, but to give a number, just to give you...
The duty cycle is only 5%.
Sure. No, my point was to say that to compare this force, this 100 piconewton force to a molecular motor, this would suggest that 10 dozen of motors working together, would yes, just to give a note.
Is that roughly consistent with the measured density of myosin at the cortex?
I don't have the numbers. Yeah, so I don't have but no, I don't have.
One thing you could compare to is the contractility measurement that Eva has done.
But we never have absolute numbers.
Yes, but I think we could have with process correction spectroscopy.
So you attribute this force tension to contractility. And what about adhesion? So is it negligible or?
No. Here I do not make any assumption about the origin of this tension. So here I really measure the tension by this. I do not know. Then it would be important to know when you change the organization of actin networks, when you increase the level of scattering. Then it's, the hard work starts as well. But it's here I just say there is tension of the range is 100 piconewton, and there is a twofold difference. And just to give a partial answer, so we've just started to do experiments where we inhibit, so we sequester actin monomers and depolymerize actin by latrunculin A, and we do see a difference in terms of tension. But this has to be completely... Yes. Yes.
So that would suggest that maybe the residual tension that you have in the lat A experiment is one contributed by, say, membrane elasticity.
It might be. I think we have to improve this. We have to do more of these experiments too.
Did you try to go longer time chains or longer time chains?
We have started, but I'm sorry, I have not more data to show. So at least with quantities. What I would like to show you is that not only we can measure tensions at cell boundaries, but as well we can address questions, I think, which are interested in the field of tissue morphogenesis. In tissue morphogenesis, which is when, if you think of molecular motors pulling at cell boundaries, how far forces will produce deformation, and to convince you that we might be able to answer this question, here is a view of more cells in the tissue. So you do see this guy pushing onto this membrane, on these two membranes, and if I hide just to be sure that you do see as well as I do see deformation at long range, is indeed here you have deformation where the bead moves the junction, but here as well. So there is transmission through the network of junctions. I have no numbers to give you, at least no to say how it propagates, how far, what is the
So I'm a little uncertain here of what's happening on the whole cell level, but you have one focal plane here. Presumably there's adhesion junctions in that plane. But is the rest of the cell similar?
Yes, it does matter. So indeed, if it's much easier to deform membranes if you go below the adherent junction plane. So the adherent junction plane is stiffer. I mean, the tension is higher. But it's easier. It's easier, basal.
And so they are much more.
Yes, it's easier for the same from.
If you do this,
so with the setup we have, we cannot image in a plane which is different of the plane of the trapping. There are, Next, it might be feasible, but then you have to change the focus of one beam with respect to the beam of imaging. This might, this is feasible, but we haven't done it. Okay, so what you do see indeed that there are deformation, and we have started to quantify this deformation here by using physics. Tricks of physics using synchronous detection. So if you do a Fourier image at each pixel of this movie, because you have a sinusoidal oscillation or any oscillatory movement, you can see which membranes do move during this process. So this suggests that indeed, even here you produce 100 nanometer deformation, but even at long range, you do see as well deformation of 30 to 50 nanometers. So now the question is how it propagates, how it depends on the properties of the networks, the topology even of the cell. This I don't know yet.
How this anisotropy is set up and maintained? So how does the cell know what is vertical and horizontal?
So this is not known. I'm sure that Thomas will address this question during his talk. Even if I would say the final answer, is not known. The patterning we have been that have been discussed are important for this polarity, but how this gene patterning translates into a polarity of myosin 2 is not known. Am I right, Thomas?
Yeah. We a little bit. So from this data, can we infer storage modules in the last?
Not yet. Not yet. This is the not yet. Okay. Okay, I changed gear, as I said. I will now. How much time I have? 40 minutes. Yeah. Are there questions about the first part about the mechanical measurements? Still. Yes.
How thick is the actin cable in the Z direction?
It's deep. How thick? It's I would say below 1 micrometer, less than 1 micrometer. I don't know exactly the thickness.
Lactin is probably more.
Lactin, yeah.
Yes.
Yes.
Can you do this experiment in different planes?
Yes, this I can do. And at least the first. I don't have much data, but at least it seems that it's easier to move more basally. Away from the plane of adherent junction.
Can you combine your two kinds of experiments here? And if you ablate the myosin in an adjacent cell, and then you look at the coordinated movements, the transmission of movements, can you show that the ablation of the myosin blocks the transmission of force and movement?
It's a good idea. I mean, at least I cannot do it right now, but I mean, you can combine, in terms of the method, it's not that difficult to combine the two methods. I mean, to combine the two methods. I mean, yes, to see how, yeah, local disruption of Mizing 2 can change the properties. Yes. Yeah.
Go forward.
No. Yeah. I think I still have some time to answer questions. I know that I have still 40 minutes to go, so I know what I've. Yes.
I have a question along the same line. So
about the sorry.
Along the same line. So,
Okay.
Firstly, do you see any junction remodeling during your oscillatory manipulation process?
At least I don't have enough data to answer this question. I need to get more. To know whether the mechanical whether there would be like a mechanotransduction effect, whether because you push repeatedly on this junction will drive processes. This I really don't know.
Another question is it possible to coat the bead that you're manipulating with something that will be interacting with the actual mousing network to mimic the
Yes.
mousing code?
We haven't done it, but I think it's feasible to do it and to prepare beads. We made some trials which were not successful, and we had problem of aggregation of beads before injection. But there is maybe some surface chemistry which can solve this. This. Okay. Yes.
Do you have displacement when you push on a vertex, or have you ever tried to push on a vertex?
Yes, I think I have one movie, maybe which is one of the hidden one. Here the deformation is quite small here. We are lucky to have a bead. I mean, at least with this configuration, which is quite symmetric. So you do see some changes. We try to, at least with our method, to measure how far it propagates. Okay, it's one example so far. But okay, so now the might be as well good for discussion with different theoreticians from the crowd, that how you can with different type of mode of propagation you can have, depending whether you move junction this way or that way. There are some this prediction from the theory of elasticity that would expect that if you pull normal to the junction here, you can excite different modes of propagation, which corresponds to vertices moving like that, but as well moving like that. And this has been, I think.
I don't know anything about all this, but doesn't it suggest that there's a compression resistance in your boundary? I don't understand why that.
No.
Because you're pressing up against.
But
if there was only tension involved here, why should pushing directly in line with?
In fact, what you do is you change the force balance. So it's like by pushing here, it's like if you would pour more tension onto this bond. So you change the force balance, and compression. I mean, yes, you press, but what do you mean? Is that you might expect different outcomes depending on the property of the materials? Or?
Yeah, so I mean, I don't, with the way you were talking about tension and the way I think about myosin,
okay.
I think a bunch of ropes connected.
Yeah, sorry.
And the ropes,
yes. I think your point is related to the fact that there is some constraint on the cell area, which makes that the system cannot change, can get any shape. I mean, if it would be only junction with no pressure, for example, here in the cells, then indeed you can expect that to have different types of change.
That thing's made out of concrete instead of a rope. There are 3 ropes.
Yeah, there are 3 ropes.
You're still pulling on 2 other ropes.
Yes, but if you pull on the 2 other ropes, if there's no compression resistance on the 3rd rope, and you're hitting directly in line with it, I don't see why it shouldn't bend as much as it did if it wasn't there.
Maybe we can do it in the next.
Okay, we can do it then. Okay. Okay. Stuck?
It's a mechanical engineering department. This question.
You didn't like this movie? Sorry, I'm stuck. Okay, sorry, I have to reboot. Sorry, it's stuck, so I have to reboot. Sorry?
That happens with chalk doors.
Yeah.
You can't have that movie with that lead.
I know it. Yes, not a big problem. I have to reboot because it was stuck. Sorry. And I can still answer questions maybe.
Well, now you can ask the question on the blackboard.
The question about the.
So my question, basically, I think I could rephrase my question is why not consider the fact that it's possible that these things are stiff and that there can be resistance.
They are stiff. I mean, they are stiff. I mean, they can be stiff and tensed. I mean, the two. I mean, you can have the 2 properties, which you can have a spring, which has a given stiffness, and the tension will depend how much you deform it.
Okay. And then, for me, when you got that,
yes,
you got that situation.
I agree. So the question is, if they would be completely rigid, they can be tensed. Okay, if I push here, this length will not change. Will change the angles if they are fully rigid. Now, if there is a finite stiffness, you can as well deform.
Yes.
So this then by looking how much you change the length versus the angles, you
can get both measures. And so is there significant stiffness? Are these rigid rods?
Sorry?
Is there significant stiffness? Are they like rigid rods or are they like floppy ropes that are under tension?
They do not look very floppy since when you... I mean, if they would be floppy you could deform them like by pushing. Yeah. Sorry, what happens? Takes more time than. that I have a problem with my computer. Il a rebooté 2 fois tout seul. I think I have a copy. Yeah, it's a keynote reason.
On va le laisser se reposer. Don't leave.
Very exciting. You've been trying to synthetic magnetic beads.
Yes.
Unsuccessful. Well, successful in some ways.
I think it's. It doesn't indicate. Yeah, it's there. Okay, TPS, yes. Maybe you have to put on your to be sure that. Yes, it's back. It's perfect. Okay.
This is going to take 12 minutes.
Okay.
I'll keep this copy.
Yes.
She built
Okay. Okay, so
exactly. But she could never. Do this tomorrow.
okay, it's back.
It could use mine. Actually, what if we try mine?
It's back. It's almost back. It's running like je sais pas, c'est bien. I need 2 minutes more. I mean, it's going to be fine. Sorry. I hope it's going to be fine.
Wir machen das.
Je sais pas ce qu'il fait. Attends, non, c'est bon.
Voilà, c'est bon. Là, lance-le comme ça.
Attends, il faut que je me mette là.
C'est le projecteur. À la diapo suivante.
Okay.
Ja, theoretisch ja.
Okay, so sorry for that. So it was like a long transition, a pause between two different parts of my talk. Sorry for that. So now I would like to discuss different questions still at cell contacts. And what I will be talking about is how Ikarin organizes at cell junctions during tissue morphogenesis. And so you've seen already morphogenetic changes. Some of them are driven by changes in or fluctuation or levels of Ikarin or N-cadherin. It was the case for the cone cells during jump-on elongation as well. You have fluctuation of Ikarin, which give rise or fluctuation or change in level of Ikarin, which drive cell intercalation. And what I'm interested in here is to understanding the principles which underlie Ikarin organization at cell contacts and how regulatory molecules can change this distribution, how they do it. And so at the molecular level, cadherin can form bonds, so contact between two cells by transbinding. They can as well bind in the same plane of the membrane by cis binding. And then there are cortical regulators which can change the organization, and we'll see how they can, and as well the dynamic of these clusters, as I will show you. And as well, an important aspect is how under synthesis, recycling, deposition, and removal of cadherin can change distribution and eventually shapes of cells during tissue morphogenesis. So if you look at confocal images of E-cadherin, you do see that already that E-cadherin is not distributed uniformly along contact. There are regions where there is more cadherin, patches of E-cadherin than at other points. And these structures are diffraction limited, so they are presumably below, they are below 500 nanometers in size. And these points of E-cadherin are important for adhesion, but as well because they are anchoring points for forces during tissue morphogenesis. If you look at the EM pictures, these points of adhesion appear as dense electronic structures, points of contact between cells, and are called spot adherent junctions here in the Drosophila embryo, and they have been observed in different animals. And if you look at this organization, so this is now dynamic, so movie of the movement of cadherin at cell surfaces that we've shown a few years ago. So Thomas work with Thomas show that at least these regions where there is a lot of cadherin they move very slowly, in fact, with respect to points of reference, which are in this case these vertices. And if you look at the recovery, so how fast these big structures recover, they recover very, very, very little. So if you trap the structures at cell junctions, you do see that there's very little recovery of E-cadherin, suggesting that the turnover is very, very slow. Whereas if you do the same experiment in regions where you don't have these big spots of E-cadherin, you have large recovery when you do the FRAP experiments. So we were intrigued.
Different here. Is it like the minor recovery that you see when
you see little more?
Essentially the same recovery that's happening.
So So there is two things. The time scale of recovery is about the same, but what here, but the fraction is very different. So what this suggests that when you do FRAP experiments on these spots, you have mixtures, and I will show you the mixture it is, of clusters which look like immobile structure, where you don't have recovery because the turnover is very small, and then this time scale is related to diffusion of either single molecules or small clusters.
The same thing is happening in the
yeah, I mean, still you have fast diffusing spaces. Diffusing spaces which can be monomers, many dimers. I mean, we cannot resolve, and a fraction of clusters which are more stable. Okay. They are normalized. Yeah, they are normalized. Ah, you mean this? Okay, they are normalized. Okay. Okay, so now we wanted to go. We were intrigued by these clusters, and we decided to go for high resolution. And I will explain you why we did it. Not only to get nicer images. I mean, we get nicer images, but the important aspect here is that we can count molecules and compare different genotypes, and I will show you how we do it. And what is high resolution imaging? So in the course last week, unfortunately, you haven't had a course on super resolution imaging, so I will go fast on this just to get an idea. So the basic idea of any high resolution technique, imaging technique, is based on the fact that you can localize a molecule with a better accuracy than the resolution. So the resolution tells you that you cannot distinguish two molecules when they are very close to one to another at a distance below that 200 nanometers. This is a criterion of resolution. But if you have a single molecule, if you are sure there is a single molecule, and if the signal-to-noise intensity, signal-to-noise ratio is high, then you can localize the peak of maximum with a precision which can be very high. It can be in nanometers in cases where you have very bright molecules. And so this is due to different. This method has been proposed a few years ago by different groups, BETSIG, for PALM, Chang for STORM, and this is related to this idea of pointillism painting, where you get an image from many points. And since he's not in the audience, I want to thank Binian Foucault who did the work. And again, so you have to localize molecules, and in a dense material such as a cell, there are many, many molecules. So how can you be sure that there are only single molecules at a given time? So then you use a trick. You need switching molecules, which can be off. And then you switch a small subset of all the molecules, which is them all. You localize them. You bleach them. And this is a good thing about bleach molecules which can bleach. And you then collect all the position of all molecules very, very high accuracy. So we did this, we implemented this method in vivo. When I say in vivo, in a tissue, it's fixed tissue, and you will see since you need to get all the molecules, you need to get many images. So this is the standard image you get in the microscope. In fact, it is the sum of all the PALM images. And if now you use the super localization, the localization precision, you can see that structure that you could not resolve with standard microscopy, in this case epifluorescence or oblique illumination, you can resolve with PALM. And now the structures that in confocal you do see, like, okay, maybe there is something here, something there. You can resolve E-carrying here, molecules here. It's a density map where you see the different clusters. And since you detect, okay, just for practical point of view, as I said, we do not do it in vivo, live, but still we can do it in a whole organism, and we fix the cells. And then we have to collect to get all the E-cadherin, so to count all the cadherins, we have to get 20,000 images, so it lasts 30 minutes. So if you remember the speed of movement of jambon extension, you cannot do this live with this constraint. And with our setup, we have localization precision of 30 nanometers in the plane of the tissue, and we have as well a 50 to 100 nanometers localization precision along the axial direction by using some optic trips. I think I won't have time to discuss, but if you want, I can discuss this later. So we localize the molecules, and now we have a map of the molecules, so E-cadherin molecules in epithelia. So each of these points is an E-cadherin molecule. So you can see how they're organized. So there are clusters that we can recognize. So here the colors corresponds to the number of molecules per cluster in the plane of a junction here with an oblique view. So we can now address different questions. How clusters are organized? Is it random distribution? How different regulators can change this distribution? And the first question we tried to address was, are there cis or trans clusters? To which extent? In fact, we cannot resolve directly cis clusters. We do not see like two clusters, one apart from each other. We don't have the resolution. But still, in some occasional cases, we can see separated membranes where it's possible to recognize cis clusters. So these are cis clusters, are two membranes separated. And we use this as a ruler to know what is the density of molecule inside cis cluster. So this is the distribution. So in average, there is 5,000 molecules per micrometer squared in these clusters. Now, if you compare this distribution to what we observe in all the clusters for which we cannot all resolve this separation, you do see a shift, but it's not a twofold difference. If these clusters would be all trans clusters, we would expect to see this density of 8,000 molecules per micrometer squared. This shows that there is a mixture of cis and trans clusters, which are, by simple comparison, in a 4:1 proportions. Yes?
How well do you know that you can see all your molecules?
So I will answer this question. So there are two things. So first we check that so we use EOSFP. Okay, first we use EOSFP, monomeric EOSFP1, and we check that it's an irreversible photoconvertible protein. So you count these ones only. I mean, in small proportion, two times, but this doesn't screw up the distribution we've measured. And we do the measurements, so we do the recording to the end. We do not see any molecule at the end of the experiments. This is the first answer. The second answer is that we did as well measurements in live embryos with fluorescence correlation spectroscopy to quantify the quantity in a different way, the number of molecules, EOSFP molecule and GFP molecules. And we found the same number with some errors, but we have, and the last answer will come just afterwards. In terms of the density of molecules, we can compare it to structural data, where we know the size of the molecule and the packing of E-cadherin in clusters.
In those rare cases where the membrane appears, the calculation you've made really depends on that not being some weird thing, right?
This is true. This is true. This is true. Yes, but still, okay. Okay, what time I have? Okay, so there are some, one degree of complexity, I mean, at least something which is, in our case, we don't have endogenous E-cadherin fused to a photoconvertible protein. So we have as well the endogenous cadherin, which is there. So to quantify the missing molecules, we did these fluorescence correction spectroscopy measurements to know the ratio. So I go a bit fast on this because I think this is a bit technical, but we compared the surface density that we measured by palm, so this is this, to what we measure with FCS measurements in live embryos. This is this, in this case, for the ECAD fused to EOS-FP, and we compared this to the endogenous molecule, which is about twofold more. So that indicates that the molecules we count in the palm measurements represent 30% of the total number of molecules, ECAD molecules. And if you do the, then if you translate the density of ECAD fused to EOS-FP molecules into density of ECAD, total ECAD, this is the number, so 16,000 molecules per meter squared. And then if you ask the question what it corresponds to in terms of distance between ECAD, you get about 8 nanometers, which is in agreement with structural data where people have looked by EM and X-ray about the packing of molecules, E-cadherin molecules. So this shows that we are looking at dense clusters, densely packed clusters of E-cadherin. Okay, so now I think more interesting. So we have clusters densely packed of E-cadherin, and now I would like to discuss the distribution of these clusters. And from these, as I will show you, we can answer questions about how this organization of cadherin is regulated in vivo. So here is the distribution of cluster size. So you do see that almost every cluster size is present, from monomers to few hundred of molecules. So it's a power law with a given slope. So it's a log-log representation where you see this slope and as well a cutoff above a given size. So this is the mathematical formula which you can fit to get two numbers, so the slope here and the cutoff. And power law means that it's free scale, so there is no specific scale that a cluster would be of a given size, that the mechanism would set a size of clusters. And we ask different questions. The first one is whether what we observe is a steady state and whether it changes with time. So during morphogenesis, for example, during germ bond, there is about twofold increase in average of the level of E-cadherin. And if you look at junctions, they have different surface density of E-cadherin, different number of cadherin in total, and you can compare early and late. So in late, when I say late, at the end of germ bond elongation, where there is more E-cadherin, there is in average more E-cadherin, and there are more big clusters. But if you compare now, and this is here the strength of PALM, not only to get nice images, but really to count molecules. The good thing is that because you can count molecules, you can compare distribution of cluster size for a given surface density of molecule, for the same number of E-cadherin molecules at junctions. So if you look in early embryos, you do see that the distribution of cluster size change. So there is more clusters when the surface density of E-cadherin is larger, so you change the slope. And now if you compare from at two different stages, where the average surface density of E-cadherin is increased at later stages, in fact, you do not see any difference in terms of cluster size distribution. So this shows that the cluster size distribution does not depend on the age of the junctions. and that we really look at steady-state distribution of E-cadherin molecules at junctions.
The value of the exponent?
I will come back to that. Okay, these are, this is here. So these are the values of the exponent. So the exponent, so this slope depends on the surface on the surface density. So it increases with the density of E-cadherin adjunctions, and it is below 1 minus 1.5. And I will come back to that afterwards. You can look at the cutoff, so this size above which there is no many clusters with an exponential decay, and you can look at the amplitude. The amplitude, this monomeric species is constant over a large range of surface density of E-cadherin, suggesting that monomer concentration is immune to changes of the total junction concentration. So this suggests stability of transbinding. So once a molecule has engaged into clusters, doesn't come back into monomers easily. And if you think about, so if you ask the question how fast these clusters organize, you can do at the end of germ bond ligation, where you start to have these cell division events, you can look at, with snapshots at least, look how clusters organize after the formation of a new contact, and estimate the times of the formation of the new contact. And if you look at cluster size distribution, in fact, you do see cluster quite rapidly after the formation of a new contact. After 2 minutes, you have clusters already with significant sizes. It increases along with the concentration at junctions. And in fact, even in this case, few minutes after the formation of the new contact, you get an exponent, which is the one you have for more mature junction at the same dot CT of E-cadherin. So this shows that the clusters grow rapidly, few minutes up to steady state, which changes with the surface density of E-cadherin, and the distribution adjusts relatively fast to molecules which are deposited either by molecules coming in by trafficking or by diffusion of molecules from transverse junctions. And so now I would like to discuss how we can model this. And why we did model this is because as I will show you, we can learn how clustering is regulated, and more specifically, how recycling can change the distribution of this organization, and how active processes, actin-dependent processes can change the distribution of these clusters. And the general scheme of clustering, coagulation is something not new in physics. It has been proposed about 1 century ago by Chmielewski. So here is an adaptation of this description, where you think that cluster formation is a process which is determined by how small clusters will fuse to make larger clusters. They can break up to give smaller clusters, and you have a balance of these different processes which will yield to the steady state distribution. So these are the different terms, and in addition you have a recycling term which tells you how many molecules are deposited, and how they are deposited and how they are removed. So you can look at the steady state distribution of this given by this model, where you remove this term, and you can make prediction, as I will show you just afterwards. So here, what we can address with this approach, by comparing the high resolution data and with prediction, and looking at prediction of the model, is what are the dominant processes in cadherin clustering, what of them are controlled to produce variation with organization of actin, myosin, and how endocytosis change this distribution. So the first thing we looked at is how endocytosis can change the distribution. So you can do, for example, use a mutant, DNA mutant, which prevents the fission of vesicles. And the first observation is that the quantity of Ikarin is increased, and you have as well a change in the number of clusters. And here again, we can compare junctions with the same between the between the dynamine mutant and the control, and at the same junction surface density of Ikarin. So this is in black the control. This is for Shibir in Shibir mutant, where we block dynamine-dependent endocytosis. You have more Ikarin adjunction in average, but you can look at a given surface density and see how the distribution of clusters is changed. So here you do see that the cluster size distribution is first not changed at small sizes. This suggests already that the endocytosins does not play any role in this this distribution at this size, and it plays a role at larger sizes. So by comparing really the distribution at the same surface density, we can show, and with different surface density, we can show that the slope is unchanged while the cutoff is changed. And this shows that endocytosis targets large Ikarin clusters, and it doesn't change anything into the clustering at smaller length scale.
Clusters and transclusters?
No. I mean, this is what I just say. I have an average information. I can say in average, no. I don't have enough.
In terms of endocytosis, you sort of think that transclusters are endocytosed the same way as cis clusters?
No, I mean no. At least I mean, if you look at EM data, so the endocytic vesicles emerge. I mean, in fact, they remove Ikarin from one membrane only. So how this, whether there are coordination between these events to break up transcluster, I don't know. And I don't have the resolution to look for given clusters, whether it is endocytosed, whether it's cis or trans. No, this I don't know.
Whole cluster, not individual Ikarin molecules. Because
okay, so no. Here the point is that the point I want to make is that endocytosis is not just removing Ikarin by random. If it would be,
this doesn't show it because the effect grows with the size of the.
No, but
so it would be not very efficient for. It would be subdominant for small cluster. If you remove one by one, it grows with the.
Sure, but if the endocytosis would be random, independent of the size, you couldn't explain this power.
It's a perimeter versus surface effect. So you need a large enough surface to see the effect of endocytosis.
I think okay. Yeah, I am not sure. The thing is that here we compare really at the comparison is done for the same total number of molecules, and we do see a change only above given size. It's not just changing the concentration. It's not the same effect, because we know the effect which is produced by only a change of concentration. We know that this is the change. If endocytosis would be like changing the concentration, this would resemble the wild type, where you change the concentration, you see the slope changing by, okay.
I'd like to understand a little bit the relation between cis clusters and trans clusters and the mobile fraction, right? And so, for example,
from this picture, one would conjecture that it is cis but not trans clusters that would be endocytosed. And I think you said before that cis clusters are more mobile than the trans clusters.
I didn't say that. Yeah, sorry.
You haven't said that. Throw in the hypothesis, but I think there would be a way of checking, because you can go and do your FRAP experiment with or without endocytosis and see if the mobile fraction changes.
Yes. First, I haven't said that. So what I just said is that when we do this FRAP experiment where we don't have this resolution, we FRAP many different clusters, and I don't know whether cis versus trans move faster. I mean, this I don't know. We would need to use, for example, two colors or ways to distinguish really between cis and trans.
For example, if you do it on many clusters and you start seeing some sort of bimodality.
Yeah, the problem is that the FRAP, I mean, you've seen the FRAP, you do see like a spot in which we know there are many different clusters. There are tens of clusters of different sizes. You don't have the resolution to do the dynamic experiments to, So
So I don't know very much about this, but I thought I understood that forming a cis interaction would promote and stabilize clustering because of the bringing the membranes together.
You mean trans clusters would promote?
Oh, sorry, trans clusters.
Trans clusters.
Yeah, trans clusters. Well, trans interaction would promote clustering.
Yes. Yes. Yes.
Less force required.
Sure. Yes. Yeah. Again. Yeah.
Shouldn't there be a bias then across your spectrum where
Again, we can, but at least for endocytosis, this is the results. what I want, and again, I cannot. I know. Again, I cannot resolve cis and trans clusters, and even in the model here, we consider in fact trans clusters only because we cannot resolve. So there is no way I can give information on this with this data. So.
But I think the non-trivial result, which is that when no one knows whether endocytosis plays this quantity or something else, which would be organization. That result tells you unambiguously that there is an organization within infected, which is like large clusters all of them. And if you think about adhesion and transmission of
is it known whether there's events where the trans interactions are broken up? Is that a dynamic?
But they have to be broken up when you remodel a junction. You have to remove the trans clusters for cells to integrate. So this you could do by endocytosis. If you remove one side of the clusters, and you will get a cis cluster, and then you can remodel.
Can you do it by flow?
Then it's another question. Okay.
So how about let's
Yes. Okay. So I continue. So now, and it follows up a question of Jacques. What do we learn from this exponent? Okay. So first, if the system would be at steady state and completely passive, so just molecules diffusing, interacting, short range, with no and only fusion of clusters. So you can show, in fact, that the cluster size distribution follow this slope of minus 1.5. So clusters are populated because small clusters fuse to make larger clusters, and the population of clusters of a given size is determined by the flux in and the flux out, and you populate by cascades the different cluster size. Now, imagine that you have a process, an active process, which would depend on the size, and in a power law fashion. So, imagine that you populate, so the flux in for a given size is larger than the flux out, and that for smaller clusters, this rate is faster than for larger clusters. Then, because there is more clusters fusing to give a cluster of given size, you will end up with clusters of larger size. Larger clusters will grow faster than small ones, and you will have a change in the slope of this size distribution. And now you can have the reverse process, clusters of larger size fusing faster than clusters of smaller size, and you will have a change in the other direction. And so this is a size-dependent fusion model. You can complicate the process. Now you have fusion processes, but you have as well fission processes. And if I take just this example, imagine that there is population of clusters by fusion, and if the clusters break up, the cluster of small size break up faster than cluster for large size, you will populate more clusters of small size compared to large ones. So you can change the slope by processes, by tuning the active processes, fusion or fission processes. So now if I come back to the experiments, I show you that the power law exponent, whatever the surface density of E-cadherin, is always below minus 1.5. So already we can rule out, these two classes of models that there is more accumulation at large cluster size due to fusion processes being size dependent or this way. So we can rule out these type of processes, but we are left with these different types of processes, either size-dependent fusion or size-dependent fission model. Now, how active processes regulate this distribution? And we know that actin can regulate clustering. It has been shown in different systems, and how it does it. So we did experiments where we break the links between actin and encadherin by injecting alpha-cadherin I. The first result is that you have less cadherin at junctions. And now if you compare the cluster size distribution, in alpha-cadherin I and control at the same surface density of E-cadherin, you do see a change in slope. So this shows that in this case, F-actin controls the clustering, the slope of this distribution, so promotes clustering, and this either suggests that it promotes fusion processes or change fission processes, or a combination of the two. And this is true for different density of E-cadherin at adherent junction. Again, we are left with these two different models. So imagine that the role, so what we see in alpha-cadherin I, that it steepens this distribution, that in a size-dependent fusion model, it would suggest that actin-cadherin interaction would reduce the fusion of larger clusters compared to small ones. This is rather, it's not very plausible given what we know about E-cadherin-actin interaction, whereas in the size-dependent fusion model, the role of alpha-catenin in this case would be to reduce the rupture, the breakage of smaller clusters compared to large ones. So by looking at these different effects, we can show that the coupling between E-cadherin and actin, as revealed by alpha-cadherin I, shows that the control of E-cadherin is tuned in a size-dependent manner through a fission model. So to conclude, maybe I went a bit fast on this part, and we can discuss this in the afternoon. So the model, at least with a cartoon, is the following: is that you can have a very precise control and local control of adhesion points. Of course, the level of Iketerin is important. It determines the distribution of clusters. But importantly, and you can understand this by simple physical rules, coagulation processes, fission process. But in addition to this passive mechanism, you have important active mechanisms which can change the distribution of clusters and therefore modulate the anchoring points of forces. Actin interactions reduce the fission of clusters of smaller size, and endocytosis has not only a role on the level of Iketerin, but as well on the distribution by targeting large cluster size. So this provides a nice way efficient way to modulate adhesion points and therefore modulates force transmission. So it remains to be understood how forces themselves can change this distribution. This we don't have data with this type of approach. But at least what we have observed already with actin gives us a hint about what can happen. And this was like the biological model. This was like a physical model, like a phase diagram, which says about the same thing with more numbers. So it's a phase diagram of Iketerin clustering where I sum up all the data. Most of them I showed you. So this is the wild type where you see how clustering, the power law exponent changes, and with the size, the cutoff size. And in this diagram, you can see the effects of actin polymerization or actin depolymerization. So endocytosis, as I showed you, just move the wild type in this vertical direction, changing the cutoff size. Okay, so if you inhibit endocytosis, you will have clusters of larger sizes, whereas it has no effects on the power law exponent. And actin depolymerization of reduction of Iketerin actin interaction will move the curve into this direction with lower clustering, so lower power law exponent. And with that, I think I will stop here. And I have even time to thank again people. So the last part on the model has been done by Madhav, that you probably all know now. I showed the work of Binyan on high-resolution imaging. Olivier Blanc did the work on tweezers, beads. Thomas, you know him, and this is our place in Marseille. Thank you.
Increase endocytosis? I mean, I'm asking basically because
yes.
I mean, in the inhibition case, so basically if you inhibit endocytosis, this deviation for these large clusters, it seems that it's more getting towards the power law again. So really, is that large clusters are sort of there you feel that basically in the presence of endocytosis you have these dynamics which were getting you off the steady state, and now when you inhibit endocytosis you would
sure.
get more towards the power law. So I was wondering whether if you...
We haven't done these type of experiments. No.
You can increase it when it's over.
Okay.
And if not...
There is competition between students. Okay.
Student.
Okay. You show perhaps that in a relatively large and stable cluster, you had less recovery, right? You recovered only about 30 or 40%.
Yes.
That's compatible with the idea that we have more endocytosis in these large clusters, or can we compare at all?
I think the time scales are very different. So with these FRAP experiments, at least as they have been done, you do the FRAP over a minute time scale. 1 minute, and you might have one or maybe some endocytic events, but this is probably not the way to look at how endocytosis can produce removal. These are different time scales. And I don't think that FRAP in this case would be the good way to look at how endocytosis can produce a turnover, change in quantity. So, sorry? Photoconversion, for example. Yes.
Another question about the
SHIB.
Did you check whether you have the same ratio between endocytic and EK2-EOSFP in that particular case? If for whatever reason you affected more one EK2 than the other?
No.
Which could affect the determination of the cluster size?
No, we haven't checked that. No. At least I cannot do the same experiment with the endogenous fused to EOSFP to check that. Yeah, PHGFP. No, we tried that and it's 9. Yeah, we cannot do a resolution imaging with the PHGFP version. Yeah. Okay.
Thank you very much.